Here's a question I want you to answer honestly right now before I say another word. When did you last read the fine print on a medication bottle and actually read it? Not squint at it, not hold it under a lamp, not hand it to someone else, but genuinely read it the way you could 10 years ago. If you had to pause before answering that, this video is specifically for you. Because here is what almost nobody in medicine will say plainly. Most people over 60 who are losing their vision are not losing it because their eyes are wearing out. They are losing it because something is happening in their blood and repeatedly, silently, every single day that is chemically degrading the most oxygen-hungry tissue in the entire human body, and they are being handed stronger glasses instead of an explanation. I'm going to give you the explanation today in full, the mechanisms, the biology, what is actually happening inside your eye right now while you watch this. And then I am going to give you a very specific food-based protocol that addresses each mechanism directly. No prescriptions, no procedures, things you can start tonight. Stay with me through this entire video because what I am going to describe in the final section, the protocol itself, contains one detail about meal timing that most people get completely backwards, and it is the detail that matters most for nighttime retinal repair. Before we go further, if you are new here, I am Dr. Franklin, and this channel exists for one reason: to give older adults the metabolic and physiological explanations that a standard 15-minute doctor's appointment simply does not have time for. If that is useful to you, subscribe and hit the bell. I release new content every week, and I want you to catch it. Now, before I go into the mechanisms, I need to stop and be very direct about something. There are symptoms that mean you stop this video right now and call an ophthalmologist today. If you have had a sudden loss of vision in one eye that developed over hours, if you see a dark curtain or shadow creeping across your visual field, if you have had a sudden eruption of new floaters, dozens of dark specks or strings that were not there yesterday, if you are seeing flashing lights in your peripheral vision that keep returning, any one of those is a medical emergency. Those symptoms describe potential retinal detachment or acute hemorrhage. Nothing in this video is relevant to those situations. You need same-day evaluation by an ophthalmologist. If what you have is the slow kind of progressive blurring over months, halos around headlights at night, reading glasses that keep needing updates, general visual haziness that no prescription fully corrects, then what I am about to describe is almost certainly part of what is driving it. Stay with me. Mechanism 1, the lens swells. Let me start with something that is happening to your vision right now today in real time. Something that explains a mystery that confuses millions of people who wear glasses. Have you ever noticed that your vision is not the same every day? Some mornings things are clearer, some afternoons the blur gets worse. After a large meal, bread, pasta, something sweet, things seem to go a little soft. You might have thought it was fatigue. You might have assumed it was the lighting. It is neither. Your eye's lens, the clear, flexible disc that sits behind your pupil and bends light onto your retina, has no blood supply. It cannot draw nutrients directly from your circulation the way your heart or liver can. Instead, it absorbs everything it needs directly from the fluid surrounding it, and that fluid is in direct chemical communication with your blood. When your blood sugar rises sharply after a meal, glucose floods into that surrounding fluid. Inside the lens, an enzyme converts that excess glucose into a compound called sorbitol. Here's the critical detail. Sorbitol is trapped. There is no mechanism to carry it back out. Research published in NIH Stat Pearls describes the polyol pathway precisely. Sorbitol is produced faster than it can be converted onward, and its polar molecular structure prevents it from diffusing back out of the cell. It builds up inside the lens tissue, and because sorbitol is osmotically active, meaning it aggressively pulls water toward itself, the lens physically absorbs water and swells. Think of a dry kitchen sponge dropped into a glass of water. It expands. It changes shape. A lens that has physically swollen cannot hold its focal point. Light lands in the wrong place. What was sharp becomes soft. This is not structural damage. This is a fluid shift, and it is completely reversible. But here's what makes it dangerous over time. When this swelling and shrinking cycle happens day after day, month after month, year after year, the proteins inside the lens begin to denature. They cloud. They harden. That is how a cataract forms. The temporary blurring becomes permanent opacity. A review published in the Journal of Ophthalmology identifies the polyol pathway and its resulting osmotic stress as a primary mediator of diabetic cataract formation. the same mechanism operating in anyone whose blood sugar runs high enough long enough. Your optometrist is measuring the optical result of this process and giving you corrective lenses. Nobody is measuring the process itself. Mechanism 2, the capillaries begin to fail. Now, let us go deeper because this second mechanism is the one that keeps me up at night when I think about people over 60 who have been told there is nothing to do but monitor their eyes. The retina, the paper-thin sheet of photoreceptor cells at the back of your eye that actually converts light into the images your brain sees, is fed by the smallest blood vessels in the human body. We are talking about capillaries so narrow that red blood cells travel through them in single file. These are not backup systems. These vessels are the entire lifeline of your visual system. Without them, your photoreceptors go dark within minutes. Chronically elevated blood sugar attacks those capillaries through a process called advanced glycation. When glucose molecules attach to proteins, which they do constantly at elevated concentrations, they chemically deform those proteins. The proteins stiffen. They lose their elasticity. The capillary wall, which is supposed to flex and regulate what passes through it, becomes rigid, and a rigid capillary eventually cracks and leaks. Here's the part that physicians rarely say clearly. You do not need a diabetes diagnosis for this to be happening. Research published in the journal, I found that retinopathy was already present in approximately 7.7% of adults with prediabetes in a nationally representative U.S. cohort, people who had never received a diabetes diagnosis. A 2022 systematic review in ACTA confirmed a median retinopathy prevalence of 7.1% across prediabetic populations worldwide. The current clinical thresholds for diabetes were simply not designed to catch this level of subclinical damage. You need cumulative glycation exposure. And cumulative glycation exposure is built by the full picture of your glucose life. How high your blood sugar climbs after meals, how long it stays elevated, and how many times that pattern repeats each day. Postprandial spikes are a significant contributing factor to that cumulative load. A 2020 study from the Northern Finland birth cohort found that in non-diabetic individuals, retinopathy signs were significantly associated with 30-minute and 1-hour post-load glucose levels, not just fasting values. That means what your blood sugar does after you eat matters, and it is almost never measured at a standard appointment. A person with a fasting glucose of 98, completely normal by lab standards, who eats 3 high-carbohydrate meals a day with no fiber sequencing, who sits after eating, who skips protein at breakfast, that person may be accumulating glycation damage in their retinal capillaries every single day, and no lab report will flag it until significant structural damage has already occurred. When those capillaries begin to leak, fluid seeps into the retina swells, visual signals distort. This is the biological beginning of what clinicians call retinopathy. But the process starts years before any diagnosis. The diagnosis is just when the damage has become measurable. If you're still here with me and finding this helpful, take a moment to type 2 in the comments. It lets me know you're watching and getting value from this. Mechanism 3, the macula loses its shield. There is a specific region of your retina that is responsible for everything you use your sharpest vision for. Reading, recognizing faces, seeing the details that make life feel rich and navigable. That region is called the macula, and it is ground zero for macular degeneration, one of the leading causes of permanent vision loss in people over 60. The macula protects itself with 2 specialized pigments, lutein and zeaxanthin. These pigments sit like a biological filter in front of the photoreceptors, absorbing blue and ultraviolet light before it can reach and damage the cells underneath. Think of them as the macula's internal sunglasses, built in, always on, always working. But here is what depletes them faster than almost anything else. Oxidative stress from blood sugar dysregulation. When blood glucose runs high, the retina floods with free radicals, chemically reactive molecules that attack cellular structures. Lutein and zeaxanthin get consumed, neutralizing those free radicals. They are sacrificed to protect the tissue beneath them. And when the supply of those pigments drops below a critical threshold, light hits the photoreceptors unfiltered. Cumulative damage accumulates. Macular degeneration is not a random fate. In many cases, it is a supply and demand failure. The macula's defenses being depleted faster than they are replenished. Most people over 60 are already low in lutein and zeaxanthin because their diets are low in dark leafy greens. Add blood sugar dysregulation on top of that, and the depletion accelerates dramatically. The protocol I'm going to give you addresses this directly with specific foods and a specific preparation method that most people completely get wrong. Hold that thought before we begin. Take a second and tell me your name and where you're tuning in from. I go through the comments and I genuinely want to know who I'm speaking to and supporting here. Mechanism 4, the pressure builds inside your eye. This fourth mechanism connects your vision to something you have probably never thought of as related to your eyes, your kidneys. The eye and the kidney share a unique biological feature. Both organs rely heavily on a particular type of tiny vessel that is among the most glycation vulnerable in the body. This is not a coincidence of anatomy. It reflects a shared vulnerability, which is why in people with long-standing blood sugar dysregulation, kidney function, and retinal function tend to decline in parallel. But the kidney-eye connection runs even deeper than shared vessel type. As kidney function quietly degrades, and it can degrade significantly before any symptoms appear, fluid regulation in the body shifts. Blood pressure tends to climb, and blood pressure affects the pressure inside the eye. What physicians call intraocular pressure. Elevated intraocular pressure is the primary driver of glaucoma. Glaucoma destroys the optic nerve, not the retina, the nerve itself, the cable that carries every visual signal from your eye to your brain. It takes peripheral vision first, so slowly and so painlessly that most people lose a significant portion of their visual field before they notice anything is wrong. The chain of events, blood sugar dysregulation, microvascular glycation, kidney strain, rising blood pressure, elevated intraocular pressure, optic nerve damage is rarely presented as the single connected sequence it actually is. It is treated as separate problems by separate specialists, but it is one chain and blood sugar is the first link. And if you've made it this far and you're getting value from this, type the number 4 in the comments. It's a simple way to let me know you're here and following along. I want to tell you about someone I will call Margaret, 71 years old, retired school teacher. She was not diabetic. Her fasting glucose never exceeded 104. Her doctor had told her for years that her blood work looked fine for her age. But Margaret had been getting new glasses every 18 months for the past 6 years. Each time a stronger prescription, each time a few months of clarity, followed by a slow return of the blur. Her ophthalmologist had noted early drusen deposits, small protein accumulations under the retina that signal macular degeneration risk, but said there was nothing to do but watch and wait, watch and wait. That phrase is cold comfort when it is your vision you are watching disappear. When Margaret described her symptoms in more detail, a pattern came through immediately. The blurring was noticeably worse in the late afternoon. It was sharpest first thing in the morning, before she had eaten anything. After her usual dinner, pasta or rice, sometimes a small dessert, the halos around the kitchen lights were almost disorienting. After a day of eating mostly eggs, fish, and vegetables with very little starch, she felt sharper. She had never connected those observations because nobody had told her to. Her eye doctor was measuring refraction, not glucose dynamics. Her primary care doctor was watching her fasting number, not her postprandial spikes. Nobody was looking at the whole picture. Margaret's postprandial glucose, her blood sugar 1 hour after meals, had never been formally measured. When she started tracking it with a basic glucometer, she was shocked. After her typical dinner, her glucose was hitting 178, not diabetic, not even flagged as pre-diabetic by most guidelines. But running that high multiple times a day, year after year, over 4 months of following the protocol I am about to describe, her afternoon blurring significantly reduced. The halos at night became manageable. On her next ophthalmology evaluation, her drusen had not progressed. The damage already done did not reverse. That is not how biology works. But the environment driving new damage had changed. And for Margaret, that was everything. Mechanism 5. The nutrients are gone. The fifth mechanism is the most immediately correctable, and it is the one most overlooked by everyone. 3 nutrients are specifically critical for retinal function and specifically depleted by the metabolic conditions we have just discussed. Thiamine, vitamin B1. Thiamine is the key that unlocks glucose metabolism in neural tissue. The retina is functionally neural tissue. It is an extension of the brain. When blood sugar runs high chronically, thiamine is burned through at an accelerated rate, simply managing the glucose load. A peer-reviewed study published in the journal Diabetes found that high glucose conditions impair thiamine transport inside retinal cells and through the inner blood retinal barrier, meaning the retina cannot get the thiamine it needs, even when there is enough circulating in the blood. When the ganglion cells of the retina, the cells that collect and transmit visual signals, are thiamine depleted, the signal degrades. Vision becomes less crisp, less sharp, less contrasty. The eye looks structurally intact on imaging. The problem is biochemical. Magnesium. Magnesium is involved in over 300 enzymatic reactions in the human body, including the processes that regulate blood vessel tone, protect neural tissue from oxidative damage, and critically govern insulin signaling. Magnesium deficiency makes blood sugar dysregulation worse. Blood sugar dysregulation depletes more magnesium. It is a loop compounding in one direction, and the majority of people over 60 are insufficient in magnesium without knowing it because magnesium is not on a standard metabolic panel. Zinc. The retina has one of the highest zinc concentrations of any tissue in the body. Zinc is essential for the visual cycle, the biochemical cascade that converts incoming light into the nerve signal your brain reads as an image. It is also central to the antioxidant enzymes that protect photoreceptors from oxidative damage. Zinc absorption declines with age, particularly as stomach acid production decreases, which affects the majority of people over 60. When zinc falls low, the visual cycle slows. The retina's defenses weaken at the exact moment the oxidative burden from blood sugar dysregulation is increasing. 3 nutrients, all depleted by the same metabolic process, all replenishable through specific foods. Here is what to do about it. This is the protocol structured by time of day. Morning. Begin the day with protein and fat before you touch any carbohydrate. 2 eggs, whole eggs cooked any way you like, with a portion of smoked mackerel, sardines, or full-fat plain yogurt. If you prefer something lighter, a small handful of walnuts with the yogurt works. This is not about eliminating carbohydrates from your morning. It is about establishing your metabolic baseline before glucose enters the picture. A protein and fat first morning meal significantly blunts the early glucose spike and reduces sorbitol accumulation in the lens during the 2 hours after eating. The morning glucose response sets your metabolic pattern for the entire day. If you spike hard in the morning, you are running elevated for most of your waking hours. Start it flat. Now for the lutein and zeaxanthin piece, and this is the detail most people get completely wrong. Add dark leafy greens to your morning or early afternoon meal. Spinach, kale, or collarded greens. Whether you eat them cooked or raw matters less than what you eat them with. Both forms deliver lutein to your digestive system. What determines whether that lutein actually reaches your macula is fat. Lutein and zeaxanthin are fat-soluble carotenoids. Research consistently shows they require dietary fat present in the same meal to be absorbed at all. Without fat alongside them, they pass through your digestive tract without being meaningfully taken up. Add a teaspoon of olive oil or a small knob of butter to the greens when you cook or dress them. That fat is not optional. It is what turns the greens from decoration into medicine. Daytime. The central practice for every meal from now on is this: fiber and protein first, starch and sugar last. Eat your vegetables and protein at the beginning of the meal. Put the bread, the rice, the fruit, the sweet item at the end. This simple sequencing change has been studied in multiple clinical trials. In a controlled study from Weill Cornell Medical College, published in the journal BMJ Open Diabetes Research and Care, the incremental glucose peak and glucose area under the curve were both reduced by over 50% when protein and vegetables were eaten before carbohydrates compared to the reverse order. A 2025 study published in Diabetes Care by the same research group confirmed that carbohydrates' last food order improved time and range and reduced glycemic variability as measured by continuous glucose monitors. The same food, different order, dramatically different impact on your retinal capillaries. After your 2 largest meals, walk for 10 minutes. Not a workout, not a gym session. Walk around the block, walk to the end of the street and back. Muscle contraction during and after eating pulls glucose directly out of the bloodstream into working muscle tissue through insulin-independent pathways. A 2025 study published in Scientific Reports found that a 10-minute walk taken immediately after eating produced a significantly lower glucose peak compared to both a 30-minute walk taken later and complete rest, making it uniquely effective at blunting the post-meal spike. A 10-minute walk after a meal is the lowest cost retinal protection available to anyone, and it requires nothing except a pair of shoes and a willingness to move. Evening, the nutrient replenishment window, a small handful, about 30 grams of raw, unsalted pumpkin seeds in the late afternoon or with dinner. Pumpkin seeds are among the most zinc and magnesium-dense foods available at any grocery store. And in their raw, unprocessed form, those minerals are in a matrix the body absorbs efficiently. This is not a supplement, it is food. The difference matters. 2 to 3 servings of fatty fish per week, mackerel, sardines, or herring. These provide thiamine alongside omega-3 fatty acids that reduce systemic retinal inflammation and directly support the structural integrity of photoreceptor cell membranes. A standard portion at dinner 2 to 3 times a week is sufficient. 2 egg yolks per day. The yolk, not the white. The yolk contains lutein and zeaxanthin in the most bioavailable form of any food we know of. More bioavailable than dark leafy greens. More bioavailable than most supplements. A controlled crossover study from the USDA Human Nutrition Research Center at Tufts University, published in the Journal of Nutrition, found that serum lutein response was significantly higher after egg consumption than after spinach or lutein supplements, providing the same dose because the fat naturally present in the yolk matrix ensures complete absorption. The concerns about egg yolks and cardiovascular risk have been substantially revised by the research of the last decade. For the purposes of this conversation, the relevant fact is simpler. Egg yolks are the single most efficient macular pigment delivery system at an ordinary grocery store. 2 per day before bed. The detail that most people get completely backwards. Stop eating at least 2.5 hours before sleep. The last food of the evening should be finished well before your head touches the pillow. Here is why this matters more than almost anything else in this protocol. Your photoreceptors, the rods and cones that converted light into signal all day, follow a continuous daily renewal cycle. Research published in the journal Cells confirms that photoreceptor outer segments are in a constant state of regeneration. Old membrane discs are shed from the tips, and new ones are built at the base, following a circadian rhythm tied to the light-dark cycle. This renewal process is your retina's primary maintenance window. It is metabolically demanding. It requires resources, quiet, and low oxidative load to run properly. When you eat close to sleep, blood glucose rises during the first hours of the night. That glucose excursion floods the retinal environment with the same oxidative stress we have been discussing at exactly the window when the renewal cycle is running. You are adding chemical noise to a system that needs silence to do its repair work. You are undermining the process. Stop eating 2.5 hours before sleep. Give the retina its recovery window by giving your digestive system its rest window. This single habit change may be the most impactful thing in this entire video. You do not need a glucometer to start noticing what is happening in your own body. Here are 2 observations you can begin right now. Observation 1, the time of day test. For the next 3 days, simply note when your vision is sharpest and when it is most blurred. If blurring reliably worsens 30 to 90 minutes after a starchy or sweet meal and is noticeably sharper first thing in the morning before eating, that is the lens swelling pattern. That is blood sugar driving the blur. That is not permanent structural disease. It is a metabolic signal, and it responds to metabolic change. Observation 2, the 7-day meal log. For 1 week, write down what you ate and note your vision quality 1 hour afterward. 2 words is enough, clear or blurry. Most people who do this for 7 days see a correlation their eye doctor never asked them to look for. Because eye doctors, by the structure of their appointments, are not asking what you had for lunch. How long have you been noticing changes in your vision? Write it in the comments. A year, 5 years, longer. I read every one of them. The responses shape what this channel covers next. And the more I understand the timeline of what you are experiencing, the more specifically I can build content that addresses where you actually are. Before we close, 5 things you can do starting tonight. 1. At your next meal, eat the vegetables and protein first. Put the starch or sweet item last tonight. 2. Finish eating at least 2.5 hours before you go to sleep. If that means moving dinner earlier, move dinner earlier. 3. Tomorrow morning, cook a half cup of spinach or kale with a teaspoon of olive oil and eat it alongside 2 eggs. That is breakfast. That is also a macular protection protocol. 4. Buy raw, unsalted pumpkin seeds this week, a handful in the afternoon every day. 5. Walk for 10 minutes after dinner, not tomorrow, tonight. The prescription you keep updating is measuring a symptom. It is not addressing the cause. The cause is a chemical environment created by the foods you eat, the timing of those meals, and the metabolic state your body runs in for the majority of your waking hours. That is slowly degrading the finest blood vessels and the most metabolically demanding tissue in your body. That environment is not fixed, it is not your fate, it is a set of daily inputs, and those inputs can change. Not with a procedure, not with a stronger lens, with the sequence of a meal, a handful of seeds, 2 egg yolks, and 10 minutes of walking after dinner. Your eyes are not the story. They are the last page. Everything that brought you to this moment was written in your blood over years in a language no eye chart has ever been designed to read. Now you know how to read.